Hey y'all, this is Blasky. Last video of Unit 7, we're going to talk about titrations. Now, there's a lot of information in here. I'm going to try and get through it as quickly as I can, but this might be a little bit longer of a video than we've had the last couple days. So everything that we've been talking about so far has been all about combining different chemicals that act like acids or bases. When we talked about buffers, we started to combine a weak acid or a weak base with a salt that acted like an acid or a base, its conjugate. Today, when we talk about titrations, this is very different. Titrations are a very measured, very specific reaction between an acid and a base. So we don't have necessarily water in here, so we're changing up what we're mixing together. Three main methods that we're going to react things together. We're going to react a strong acid with a strong base. We're going to react a weak acid with a strong base. And then we're going to react a weak base with a strong acid. We are not going to worry right now in this class about reacting weak acids with weak bases. So first up, basically everything we're going to be talking about titrations is relating the amount of chemical that we've added to what the pH of that mixture is. So if we talk about our first type of titration where we have a strong acid reacting with a strong base, in this left-hand graph, we see at the very beginning of our reaction, we have a very low pH, which means that we have a strong acid. As we slowly add in sodium hydroxide, we can see that as we add in volume of sodium hydroxide, slowly that pH is going to increase a little bit, not super drastically, and then all of a sudden, wha-bam, the pH changes amazingly drastically with probably just a drop or two of sodium hydroxide. So that almost vertical section of our graph is the part that we really care about. That's where the most interesting things are happening. So there is this point on your graph called the equivalence point. So at the equivalence point, the moles of acid equals the moles of base. So we have equal amounts of moles of acid and base that have reacted with each other. When we have a strong acid and a strong base, that equivalence point happens at a pH of 7. Because it is a strong acid and a strong base, the way that they react, they're going to go through a double replacement reaction. For example, for this one, HCl and sodium hydroxide, they're going to make water and sodium chloride. Now that sodium chloride is not going to affect the pH at all. Making some more water is not going to affect the pH at all. So the equivalence point where all of that acid has reacted with base and we have not added any extra base yet happens at a pH of 7. Now, after we hit that equivalence point, any extra base that gets added in, all it's doing is just making that water solution more basic. There is no more acid left for it to react with. And so we see a very quick increase of pH up until we hit our maximum pH, where pretty much the entire solution we consider to be base. The graph on the right shows the opposite reaction, starting with sodium hydroxide and then slowly adding in a strong acid. So we start at a high pH because we're starting with a base. We slowly add in the acid, drip, 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 drip. All of a sudden, that one last drop makes us reach that equivalence point where the moles of the acid and the moles of base are equal. Again, that equivalence point is going to happen at a pH of 7. So just by looking at the shape of the graph, we can tell what's happening. If I see that that S curve is going up, I know I started at a low pH, I started with an acid. If I see that S curve coming down, I know that I started with a base because I started with a high pH. weak acids reacting with a strong base, we have a similar shaped curve with a few small differences. So when we have either a weak acid or a weak base reacting in the first place, we don't have a nice smooth S curve. We've got this little hump at the beginning. And then it flattens out a little bit, and then we get our fast increase in pH, and then it flattens back out. So still our equivalence point is in the middle of that very quick vertical section. We'll talk about that in a second. We also have this point called the half equivalence point that we are going to discuss. So I broke this down into 4 different zones for this graph. So zone 1 is just the very beginning of the reaction between the weak acid and the strong base. Zone 2 is right where it flattens out in the middle here. We're going to call that the half equivalence point. Zone 3 is here at the equivalence point, and then zone 4 is here post equivalence point. So in zone 1, the pH right before our titration begins, all that is a weak acid. So we can always figure out the pH at the very beginning of the reaction just by doing a Ka expression. 100% of the solution in there is going to be that weak acid. We will know the Ka for that weak acid. We will know the concentration of that weak acid. We can then find the H plus and the pH at that point. At zone 2, where that flattens out a little bit in the middle, we are actually forming a buffer solution at that point. Because at that point, part of that acid has reacted with some base. So some of it has donated away its proton to the base. And some of it still exists in its acid form because it's a weak acid. It's not 100% dissociating. So in that beaker at that point, you have some of the weak acid and you have some of its conjugate ion, which is exactly what a buffer is. You're not going to have to do any specific math calculations at that point. We could, but we don't have to. But what we need to recognize is there's a point called the half equivalence point. So this is where we have added in half of the volume of base that we need to get to the equivalence point. At that exact half equivalence point, the weak acid is equal to the amount of the conjugate base we have formed. We have reacted half of that acid, which means that the H3O plus or the H plus concentration is equal to the Ka, which also means that the pH is equal to the pKa. So at that very specific point, we know that the pH is equal to the pKa because this term has dropped out to 0 of our Henderson-Hasselbalch equation. That is a very common question to pop up on, especially on a multiple choice question, where you might have to look at the graph, find out what the pH is at that point, and then you can determine what the Ka of that acid is from that pH by doing a little bit of anti-logging. Zone 3 is at the equivalence point. The pH at the equivalence point for a weak acid with a strong base is going to be above 7. So the pH at the equivalence point for a weak acid plus a strong base is going to be greater than 7. Because of the way that those two chemicals are reacting, because it is a weak base, in order to get to the point where 100% of that acid has reacted with 100% of the base, we need to go slightly basic pH. We need some more hydroxides in there to react with that because each hydroxide has to pull a hydrogen off of that acid. Again, the math at that point is not super important, but being able to recognize, is that pH going to be greater than, less than, or equal to 7 is very important. At zone 4, beyond the equivalence point, the only thing that is determining the pH at that point is the excess base that you are adding in, so that we know if we've added in if it took us 50 milliliters of sodium hydroxide to get to the equivalence point, and now we've added in a total of 60 milliliters, that is 10 milliliters of excess sodium hydroxide, we could figure out what pH that's going to affect us. If we go the opposite direction, a weak base with a strong acid, again, we're going to get that opposite shape to our S-curve. So here's our funky little bump right at the beginning. We could find the pH at the very beginning by finding the Kb of that weak base and doing a Kb expression. Again, at our equivalence point for this, the pH is going to be less than 7. We're adding in that strong acid, so our pH is going to be slightly acidic at the equivalence point. At the half equivalence point, our pH equals our pKa. And then past the equivalence point, the only thing influencing this is going to be your strong acid. So that pH is going to drop quickly and stay low. Thinking about all of those things, we need to be able to figure out, well, if I'm mixing these things together, if I'm slowly dripping this strong acid into a base, how do I know what's going on inside of that reaction? One way to do it is to use pH probes. So that will generate those pretty nice curved graphs that we see. Another way to do it is to choose an indicator. So an indicator is a special chemical that changes color at a very specific pH or a small pH range. It has to do with some rearranging of the structure of that molecule, that when it's in an acidic pH, structure functions one way. When it's in a basic pH, the structure functions a different way, or things like that. One of the most common pH indicators that we're going to use is called phenolphthalein. Phenol, P-H-T-H-A-L-E-I-N. It is a totally funky looking word. Phenolphthalein. Phenolphthalein. Phenolphthalein is clear when it's in an acid, if our pH is less than 7, and it is pink if our pH is greater than 7. So that's a very nice indicator to use if we're trying to figure out whether something is acidic, basic, or neutral. We say that the point at which that indicator changes color is the end point. So we want to try and get the end point of that indicator as close to the equivalence point of our titration as we can. Remember, the equivalence point is where the number of moles of acid equals the number of moles of base in our titration. So careful and useful selection of your indicator to match where you know that pH is going to change is a useful thing. So for example, if I have a strong acid reacting with a strong base, an indicator such as phenolphthalein that changes around 7 would be fantastic. If I have a weak acid reacting with a strong base, maybe its pH is going to change at 8 or 9 or 10, and phenolphthalein might not be the best idea at that point because it's going to change colors before we actually hit that equivalence point. We are going to walk through this process in a lab in class to see how all of this works. So something else that is a common question for AP to ask is about what chemicals exist at what points in a titration. So at the very beginning of titration, I don't have a letter here, but at the very beginning, this is all acid. As we start to add in some of our strong base to the solution, at point A, we're going to have all acid still and no base. Some of that acid will have reacted with some of that base and left behind some A minus. So at point A, it's mostly H plus, a little bit of A minus. At point B, remember we call this our half equivalence point. At our equivalence point, we added in 100 milliliters of base, so our half equivalence point is where we've added in 50 milliliters of base. At that point, our acid concentration is exactly equal to its conjugate concentration. So if we were using acetic acid, Our acetic acid concentration would be exactly equal to our acetate ion concentration. At point C, at our equivalence point, all of the acid has been neutralized by base, so I do not have any acid. I also do not have any OH. All of it has reacted together, the H plus the OH to make water. At that point, we are only going to have the conjugate of the acid. Once we move past that point, that equivalence point, we're still going to have all of the same A minus that we had before, because it's not going to be able to react with anything. It's just sort of hanging out like a spectator ion now. But now we're starting to add in some excess base. It doesn't have any more acid to react with. So at point D, we now have some excess hydroxide. And then at point E and beyond, the majority of the chemical species in that mixture are going to be excess hydroxide ion. This is also a very common question to show up about acids and bases, about which substance has the highest concentration at this point along our reaction. So in order to save a little bit of time, I'm not going to do any of the example problems with you today on the video. We will do a lot of these in class over the next couple of days. So make sure that you read through all of the other information in this section, try out a couple of the example problems, try out a couple of the multiple choice, now that we've gotten through all of the content, and we will keep practicing in class. As always, if you have any questions, let me know, and I'll see you all later. Bye.